In this video, I want to talk about the electron transport chain and the FO-F1 ATP synthase. We've been working our way through cellular metabolism. We've seen how cells can take up glucose, break it down to pyruvate using glycolysis, import it into mitochondria, run the TCA cycle, and now it's time to look at the electron transport chain and the ATP synthase. Well, just as a recap of what we've learned so far, when we take glucose and we break it down, we end up with a number of products. We make a small amount of ATP and GTP, but the major products are the reduced forms of two energy carriers, NADH and FADH2. Remember, NADH is made in glycolysis, in pyruvate oxidation, and more in the TCA cycle, and that's also where the FADH2 is made. And so these substances accumulate in the matrix, where now it's time to get some energy out of them. What we're going to do now is we're going to zoom in on this part of the mitochondrion so I can show you the players that are involved. Just to orient you, I've added labels for the outer mitochondrial membrane, the inner mitochondrial membrane, and then the cytoplasm, intermembrane space, and the matrix. And now I'm going to add the components that are involved. There are two parts to this process, and together we're going to refer to them as oxidative phosphorylation. The first part is the electron transport chain, and then the second is the FO-F1 ATP synthase. And they're both located in the inner mitochondrial membrane, and they work together, but we'll see that they can be decoupled. The electron transport chain consists of four large complexes. They're called complex 1, 2, 3, and 4. There are also two smaller mobile electron carriers called coenzyme Q and cytochrome C, and I've added them here to the picture, and I want you to notice that coenzyme Q is in the inner mitochondrial membrane. Cytochrome C is in the intermembrane space, where it's closely associated with that face of the inner mitochondrial membrane. The FO-F1 ATP synthase is a sort of lollipop-shaped structure, and again, it's also found in the inner mitochondrial membrane. Okay, I want to talk about these one by one, so I'm going to make a little bit more room, and I'll move the ATP synthase out of the way for now. The complexes making up the electron transport chain are really big, and they're very complicated. They're made up of many different protein subunits. There might be a couple dozen different proteins that come together in order to form one of these complexes. And within each one of the complex, there are a number of electron carriers, so that they're way more complicated than these little block figures that I'm showing you. But just for the general overview, I'm going to simplify it this way. Here's what I want you to know. Two of the complexes can accept electrons. Complex 1 accepts electrons from NADH. Complex 2 accepts electrons from FADH2. So we've got NADH going into complex 1, FADH2 into complex 2. Three of the complexes, 1, 3, and 4, transport protons across the inner mitochondrial membrane, and they'll move them from the matrix into the inner membrane space. As they do this, it's going to generate a large electrochemical gradient, so that protons really would like to move from the inner membrane space back into the matrix. Finally, you might be wondering about the word oxidative. Well, that reflects the fact that the ultimate electron acceptor for the electron transport chain is molecular oxygen. It's going to accept two electrons from complex 4, combine them with two protons from the matrix in order to form water, the final product. Alright, well now let's switch and look at the phosphorylation side of things. And to do that, I need to take a little bit more space, and we're going to be focusing on the FO-F1 ATP synthase. This is a complicated structure, and again, it's made up of multiple subunits. The nomenclature here gets really ridiculous, with the parts that are involved in the FO being given little letter names, whereas the parts in the F1 being given Greek letters. So there'll be a combination of A and B and C subunits, as well as alpha and beta and delta and epsilon. It gets very complicated. For now, I'm going to try and keep it simple. The FO is a ring structure. It's in the inner mitochondrial membrane, and it's going to allow protons to move down their electrochemical gradient back to the matrix. As the protons move, the ring itself will rotate like a turbine engine. The F1 extends into the matrix. It's a ball-shaped structure. It looks a little bit like a tangerine with 6 slices. The major part of F1 is composed of 3 alpha and 3 beta subunits, and they're arranged so that 1 alpha and 1 beta subunit form an ATP synthesis site, and then there are 3 of them. As the FO ring springs, a shaft that extends into the interior of the ball spins, and it propels the assembly of ATP from ADP and inorganic phosphate. So the reason that we call this phosphorylation is because ADP is getting phosphorylated to form ATP. Usually, the electron transport chain and the ATP synthase work together, and we say that they're coupled. There are some drugs, however, that can uncouple the two parts of oxidative phosphorylation. For example, if we added a channel that allowed protons to move through the inner mitochondrial membrane and bypass the FO-F1 ATP synthase, the electrons could pump, but no ATP would be generated. It's even possible to isolate the FO-F1 ATP synthase, put it into artificial membranes, and then apply gradients of protons and run the pump either forward or backward in the absence of the electron transport chain. So we can manipulate these things experimentally. In addition to having exogenous factors like drugs, or researchers isolating and then manipulating these players in some kind of experiment, you should know that cells sometimes intentionally insert uncoupler proteins that will allow protons to move through the inner mitochondrial membrane in the absence of producing ATP. That'll probably strike you as being wasteful. Under those circumstances, the cell won't produce any ATP, but because it's running a futile cycle of moving electrons through the electron transport chain and then pumping protons, and because each one of those steps isn't 100% efficient, a lot of energy is given off in the form of heat. And so small animals who want to maintain their high body temperature in winter will insert uncouplers into the inner mitochondrial membrane in order to generate heat and maintain that body temperature. The protein structures for the 4 electron complexes, as well as the FO and F1 ATP synthase, are well characterized, and I'll provide some links to some excellent animations alongside this video. What I want to do in this video, though, is talk about a different way of visualizing the flow of electrons through the complexes in the inner mitochondrial membrane, and this is going to be how a chemist might draw an electron transport chain. What I'm showing you here is a reaction series, and we'll plot the various components of the electron transport chain as a function of their redox potential on this graph. I just want to remind you that from chemistry, you should know that electrons in redox reactions will move from a donor with a lower redox potential to an acceptor with a higher redox potential. We define that redox potential in terms of volts. So let me show you what I mean by this. For NADH and NAD+, for that redox pair, the redox potential under standard conditions is minus 0.32. So this is a relatively negative number, which means that NADH is a great electron donor. It'll be easy for it to give those electrons away. In contrast, oxygen down here and water, that redox pair, that has a redox potential of plus 0.82, which is very positive. And what that means is it'll be a great electron acceptor, but it might not be very good at donating its electrons. Now, although it would be convenient for us if NADH simply transferred the electrons directly to oxygen, that's not what happens. We've got these different carriers, so let me add those. So here's complex 1, complex 3, complex 4, and then coenzyme Q and cytochrome C, and I've tried to insert these plots about where their redox potentials would lie. And what I want you to notice is that NADH can hand off its electrons to complex 1, because complex 1 has a slightly more positive redox potential than does NADH. Complex 1 can transfer electrons to coenzyme Q. That has a redox potential of plus 0.045 volts under standard conditions, and that's more positive than the last of the electron carriers in complex 1. And this will continue with coenzyme Q transferring electrons to complex 3, from there to cytochrome C, from cytochrome C to complex 4, and ultimately then to the final mid-electron acceptor. Now you might have noticed that I didn't include complex 2 or FADH2 on this graph, and I'm going to do that now. FADH2 and FAD, that redox pair, under standard conditions, the redox potential is positive 0.03. And now we've got a problem, because FADH2 cannot hand off its electrons to complex 1. That would violate rules of chemistry. So instead, FADH2 will transfer electrons to complex 2, and then complex 2 can transfer them to coenzyme Q. And just notice that coenzyme Q's redox potential of 0.045 volts is sufficiently more positive than FADH2's. This is now thermodynamically viable. Now this plot shows us something else. Whenever there's a big drop in the redox potential, that corresponds to a release of energy. And so you can see that there's quite a bit of energy associated with the movement of electrons through complex 1, complex 3, and complex 4. But the nearly horizontal line for FADH2 flowing through complex 2 means that there's very little energy released during that reaction. So now if we circle back to this figure, we can look at these three take-home points and see why the electron transport chain must work in this way. So remember, complex 1 accepts electrons from NADH, complex 2 accepts them from FADH2. This has to be this way because of the energy limits. Another thing I told you was that 3 complexes could transport protons across the inner membrane space, 1, 3, and 4. Well, those are the 3 complexes that had a lot of energy associated with the movement of electrons through them. Complex 2 doesn't transport protons, and we wouldn't expect it to. There isn't enough energy to pay for that movement of protons against its electrochemical gradient. And the final point is this: coenzyme Q can accept electrons from either complex 1 or complex 3. So as it moves on to complex 3 and complex 4, what that means is all of those electrons will ultimately converge on a single electron acceptor. So oxygen will work as the electron acceptor for electrons whether they're coming from NADH or whether they were donated by FADH2. Because the electrons from NADH and FADH2 take slightly different paths through the electron transport chain, you'll sometimes see electron transport depicted as it is in this picture, where we have one pathway for NADH, that's on the top membrane with 1, 3, and 4, and a different pathway for succinate entering complex 2, going to 3 and 4. Note, however, that the reduction of both NADH and FADH2 will move protons across the membrane, and that will generate the gradient needed to run the ATP synthase.